Okay, let's go through the visual image, which talks about how the nervous system is arranged. So gray matter is made up of cell bodies in the peripheral nervous system. Nerves are bundles of axons. Receptors for touch, pain, heat, so on, carry information back to the central nervous system. Those are called afferent impulses. Central nervous system has the gray matter on the surface of the brain. There are particular accumulations in the brain, such as the visual cortex, the auditory cortex, the gustatory cortex. Nuclei are when you have cell bodies in the interior. Tracts carry information to and from the central nervous system into the brain. Columns are tracts that have a distinct location and separation to form a functional unit. So ascending pathways carry sensory information, afferent information to the brain. Descending pathways carry information from the central nervous system to skeletal muscles and to all of the response of your gut and bladder and lungs and kidney and those automatic functions that you don't think about that happen all the time. So the membrane potential. We could also say the membrane voltage. All cells have excess negative charge inside the neuron that's created and maintained by the sodium-potassium pump, which needs energy because sodium is pumped out of the cell to a high concentration gradient. Potassium is pumped back into the cell against a high concentration of potassium. The negative voltage or the membrane potential or the voltage, which means the difference in charge. So there's more negative charge on the inside of nerves, muscle, and other cells. So if you look at the membrane, you can take a voltmeter and have a reference electrode. Okay, we got somebody else coming in. A reference electrode on the outside. So if you take, so for instance, if you cut the leg off of a frog, dissected the sciatic nerve, it's a real big nerve in the frog, a bundle of axons. You could put that nerve in frog saline, which would have a concentration similar to the extracellular fluid of the frog, and you could insert a glass, a fine piece of glass that has its point very, very small, and you could insert that in the nerve, and you can measure that the inside was minus 70 millivolts compared to the outside because of these negative proteins that cannot move out. Only the small ions move out. So potassium leak channel, which allows potassium to move in and out, and that constant movement of potassium out and sodium in has to be reversed by the sodium-potassium pump, which takes energy, and there's these leak channels that allow sodium to go in, potassium to go out, but the sodium pump pumps potassium back into the inside of the nerve cell, and sodium is pumped out by energy. So that maintains the negative voltage on the inside of the nerve. Okay, let's go back. Okay, so when changes occur, when sodium channels open, the sodium rushes into the nerve, and that's called depolarization because the inside becomes more positive. When potassium channels open, when potassium leaves the cell, the membrane voltage becomes hyperpolarized or more negative. So a membrane potential has a concentration of sodium outside the cell and a concentration of potassium inside the cell. And there are special channels that allow sodium to rush in, potassium to move out, and those changes in voltage are how physical stimuli are recorded and how you move your muscles. Okay, so let's look at a picture here. So first, the sodium channel has to open, and you can see that at that point it's at about -70. When sodium channels open, sodium rushes in, the membrane potential of voltage goes from -70 to +30. At that point, the sodium channels close, the potassium channels open, and that opening allows hyperpolarization, and then it's a return to the resting state. So this is a measure of the change in voltage. As sodium rushes in, the change in voltage is propagated away from this nerve cell body, and that change in voltage is a nerve signal, a change in the amount of positive charge in the nerve and outside the nerve. So when we say action potential, we mean nerve spike, nerve message, action potential. Nerves are constantly sending information back and forth. So as you're looking and listening to this presentation, various colors and figures are being focused on your retina, and amazingly, we're able to see the visual cortex in the back of the brain analyzes that, and we can see it's absolutely incredible. So how does the propagation means how does the nerve signal move? In unmyelinated axons, the speed of conduction is slower. In saltatory propagation, that means the nerve signal jumps from the space between the myelinations and to the other. So what does that look like? So here's a unmyelinated axon. There's no myelin insulation, so when sodium comes in, the sodium is immediately attracted to this negative charge area, and the local currents begin to move, and that changes the voltage along the axon. And as that voltage is changed, sodium channels open, and then potassium channels open to repolarize the nerve. So you can see that it's a current flow. The movement of charge from the outside to the inside of the cell is current flow. The movement of charge is constantly moving from the positive area to the local current, and that local current causes sodium to come in when sodium channels open and potassium to go out when sodium channels close and potassium channels open. Okay, so what about a myelinated nerve? Myelinated nerves have a fatty covering produced by Schwann cells. So notice that it's called saltatory conduction because the impulse jumps from this node of Ranvier. That's what's called these gaps. So the local current very quickly moves to this point here because the sodium cannot. It only comes in where there's no myelination. So the local current is able to jump from an entry point at a node of Ranvier and move very quickly to the next opening. So the impulse jumps from node of Ranvier to node of Ranvier. Okay, we don't need that. Okay, information travels all around the nerve system by action potentials, nerve signals, nerve messages. The most important information, vision, balance, movement, is carried by myelinated axons. Okay, let's stop there for a moment, record that, stop recording.